Hello, my name is Clara Sun, and I am a second-year undergraduate student at Western University in the Medical Sciences program. I'm currently a member of the PM3 Computational Medicine Lab at Lawson Health Research Institute. Today, the topic of my presentation is Understanding Clinical Trial Observations Using Data Science. One of my research interests is kidney or renal disease. 30% of Canadians are affected by renal failure. Treatments for advanced renal failure, also called end-stage renal disease, often involves repeated dialysis. Not only is dialysis a healthcare burden, it also exerts significant side effects on the patient. Given the resource-intense nature of clinical trials, we sought out to create a complementary data science approach to further understand the manners in which diseases progress. In this study, I present a newly developed scientific model. This model was developed by the PM3 team in support of Lawson's clinical trial efforts led by Dr. Chris McIntyre. Human circulation is an intricate process regulated by many biophysical factors. A common treatment for patients suffering from renal failure is hemodialysis. In a typical hemodialysis session, a patient's blood will be filtered using an external dialysis machine, which contains a fluid called dialysate, and the filtered blood will then be returned into the patient's body. The fluid dynamics of a dialysis machine are shown on the left. The hemodynamic predictive mathematical model of human circulation coupled to a dialysis machine or a dialyzer is shown on the right. In our hemodynamic model, the dialyzer is integrated into the peripheral circulation. This model was used to investigate the mechanisms underlying our clinical trial observations. The objective of this study was to use hemodynamic mathematical relationships in human arterial vasculature. We expected that cardiac output in the hemodialysis population would be different from that of the control. To conduct simulation using our hemodynamic model, four model populations were simulated. These included baseline, B in gray, therapeutic hypothermia, TH in blue, hemodialysis, HD in red, and simultaneous hemodialysis and therapeutic hypothermia in green. Each of the model populations are represented by a different color. All model parameters were randomly sampled using Gaussian distributions. The hemodialysis population was generated by activating the clearance of toxin small molecules in the blood and using a hemodialysis induced increase in heart rate to 90 beats per minute. Two model conditions that were implemented independently were atrial fibrillation and exercise. Atrial fibrillation was simulated by assigning diastolic values to the left and right atrial elasticity, and also by making the left ventricle heartbeat irregular using a description called pink noise. Exercise was simulated by increasing the ventricular and systolic elastance and increasing heart rate while reducing the compartment resistances and large vessel compliances. All simulations were conducted in silico. The model was first prototyped in MATLAB and subsequently translated using a C language-based solver to generate sufficiently large model populations. Next, I will discuss results relative to the baseline values, which are shown in this figure by the gray graphs. In the four the 6 clinically relevant outputs or measurables that we identified were cardiac output, heart rate, diastolic pressure, systolic pressure, aorta shear, and kidney shear. Control cardiac output was 6,640 milliliters per second. Under non-atrial fibrillation conditions, as shown on the previous slide, hemodialysis increased cardiac output by 3% compared to the control. However, under atrial fibrillation conditions, hemodialysis reduced cardiac output by 5% compared to the control. The results for exercise were also normalized relative to the baseline population under normal conditions. We found that cardiac output increased by 15% relative to the control, and under hemodialysis conditions, in hemodialysis and exercise conditions, cardiac output increased by 18%. We also noted that under healthy control conditions, ultrafiltration was 14.3 milliliters per minute. In comparison, under renal failure conditions, ultrafiltration was observed to be 10% less efficient. We were surprised to find that exercise appeared to cause the greatest reduction in plasma volume removal. As expected, cardiac output was reduced under therapeutic hypothermia treatment. In addition, our model provided testable hemodynamic predictions. According to the model, atrial fibrillation reduced cardiac output, systolic and diastolic blood pressures, and aorta and kidney shear, but atrial fibrillation increased heart rate. These findings were consistent with clinical trial observations reported in the literature. Exercise, on the other hand, increased cardiac output, heart rate, and vascular shear. However, all of these exercise-induced augmentations were normalized by therapeutic hypothermia. This suggests that the undesirable effects of hemodialysis were partially ameliorated by TH treatment. In the future, the current model can be expanded by incorporating various feedback mechanisms such as baroreflex, autoregulation, arterial tone, renocerebral, and others. This summer, I will be working on developing a whole rat pathophysiological circulation model to assist with our ongoing experimental work. I am very grateful for support from the Kidney Clinical Research Unit and Lawson Health Research Institute. I would also like to thank the individuals who have enabled this work, including Jeremiah Joseph, Tim Hunter, Dr. Dan Goldman, and Dr. Chris McIntyre. Lastly, I would like to thank my lab and my supervisor, Dr. Sanjay Karche, for all of his guidance. Thank you all for listening.